Here at Royal Holloway, University of London, we are in quite an area where there's rich floral forage for bees. You can see it's our university campus. We have parklands here, we have suburban gardens, but what was really important for our study was that we have variation during the studies. I'm Ellie Leadbeater. I'm a Professor of Ecology and Evolution here at Royal Holloway, University of London. And my research group studies social insect behaviour, so particularly bees. And we are interested in how their minds evolve, so learning and memory. What's really nice about our setup here is that we have bees in the lab, we have an extensive social insect research facility, but we can also allow them to forage in the parkland surrounding our campus and the woodlands and the gardens. Now, you might think that memory should be something that should always be helpful for animals because it should help you make good decisions when you're out in the wild. You should be able to remember information and use it in the future. But we don't see that. We don't see that all animals evolve to have a good memory. So we wanted to work out what kinds of conditions, what kinds of environments is it that promote the evolution of memory? In other words, when is it useful? So this was a really big team effort. We carried it out over 2 years and it was led by our postdoc Chris Paul, accompanied by our technician Irina Petkova and a lot of people who did a lot of extra work.
I'm Chris Paul. I'm the lead author on this project. And so one of the unique things about our study is that we were able to control a lot of the potentially confounding variables that usually make this kind of project very, very difficult to do. And one of the great things about working with bees is that we can actually raise them all under aseasonal lab conditions here in the Bourne laboratory. And so what this means is that we're able to control all these kind of extra variables which could potentially influence our results by keeping the bees and raising them under these kind of controlled laboratory conditions before we then release them into the wild and track their foraging efficiency. So to test the memory of individual bees, we use what's called a radial arm maze, or RAM, and that's what you can see here. And essentially to solve this maze, the bee has to visit each of the arms of the maze. So the bee flies out of the entrance here, so this actually leads back to the colony, and she flies down the ends of these arms to get to the flower platform here, and you might be able to make out there's a little hole here in the wall where she sticks her proboscis, and behind that we place a tiny droplet of sugar water or nectar, but she can reach with her proboscis, and the important thing to note is she can't see which flowers are actually baited because the reward is behind the wall. So she has to rely on her memory to remember which one she's visited. And essentially to solve this task, she has to visit each of these arms, collect all the nectar which is available and return back to the colony. And the number of errors she makes while she's doing that, an error being a revisit to a flower which she's already depleted of sugar, is our measure of how good or bad her memory is. And to sort of aid them with this as well, we've printed this panoramic pictures of the lab which we placed onto the back of the wall, and so the bees can use these as kind of landmarks to solve the RAM.
So it's important to know that the bee doesn't do this just once. She does it 12 times over the course of the day, during which she learns how to use the radial arm maze. And every time she does it, we rebait the arms. It makes no sense for her to remember which arm she's visited between bouts, only within bouts. And that's why our measure is a measure of short-term memory. She has to forget when she goes back to the colony.
So each colony is tested on the RAM for 2 weeks, and then it's connected to a hole in the wall, and the bees are allowed to forage in the wild naturally on the floral resources which are available at that time of the year. And so some colonies were tested on the RAM in March, for example, and then foraged in March, whereas other colonies were tested on the RAM in October and foraged in October. And each bee, as it leaves and returns to the colony, is weighed so that we know how much nectar and pollen it collected on that trip, and so we can calculate its foraging efficiency. So we assume that floral resources are richest in spring, but how do we actually know this? So in combination with doing the rest of the experiment, we also carried out these floral surveys, again from March to October, where we assayed the generic richness of floral species across different land types. So, for example, we used quadrats and transects to count the number of genera in flower in woodland areas, in people's private gardens with their permission, and also grassy areas and landscaped areas. And indeed, what we find is that spring is the richest time of the year for floral resources, and that summer is the worst.
So remember that we hypothesized that bees with a better memory would be more efficient foragers, and we thought that that might be more obvious in the summer when resources are hard to find. Is that the case? Well, no. Actually, our results showed completely the opposite pattern. We found that those bees that did well on the radial arm maize, that had good memory scores, were the ones that foraged well in the spring, but in the summer, they were the less efficient foragers.
And remember, it's in the spring when the floral resources are at their richest, and the summer where they're at their poorest. And so this suggests then that bees with good short-term memories are better foragers in these rich, complex floral environments, but not in the summer when they're at their poorest.
So we found that memory was useful in those rich, complex environments that we saw in the spring and not in the harsh summer environments. And that raised the question of, well, why would that be? Now, we think it's to do with the fact that what we tried to capture with the radial arm maize was short-term memory. And for bees, that means the kind of memory that you use when you're foraging within a patch of flowers. So moving from flower to flower, remembering where you've been, deciding whether to leave the patch. And having a good short-term memory should help with efficiency within the patch. In the spring, you can forage within one patch, you can get everything that you need there, but in the summer, you have to fly a long way between patches. So that means that those long distances are probably dominating your flight time. And so those efficiency savings that you get within the patch are kind of drowned out by those long distances that you have to fly between them. Now, that's just a hypothesis. We don't know, we haven't tested it, but the nice thing about our study is that it opens these questions as to why it is that short-term memory is useful in a complex environment. And that's what we'd like to go on to test.